Welcome everyone to today's edition of the EV Journal Club. Our topic today, I think, is one of great importance for many diseases. Specifically, we're going to hear about how some EV subtypes can contribute to inflammation and monocyte migration. This work was recently published in JEV, the Journal of Extracellular Vesicles, as subpopulations of EVs released by TNF-α triggered human endothelial cells promote vascular inflammation and monocyte migration. So I'm also delighted today to introduce our presenter, Baharak Hosseinkhani. She's from the University of Hasselt, and so she is going to be presenting this work on which she was the corresponding and first and also co-senior author. So Baharak, thank you so much for joining us today. We're looking forward to your presentation, and I will hand the screen sharing over to you.
Thank Ken, for your nice introduction, and also I'm delighted to share our recent publications in the Journal of Extracellular Vesicles. I'm going to talk about the AV subpopulation in inflammation and monocyte migration. Why we are interested in inflammation? Because we know inflammation is a natural response of your body to a stimuli, but this good friend can become enemy when it's prolonged, and it's causing many diseases such as Alzheimer's, multiple sclerosis, autoimmune disease, and cardiovascular disease. We were focusing on cardiovascular disease because this disease is the leading cause of death in entire world. And if we look closer to the cardiovascular disease, normally a plaque is forming within the vein, and if we look closer at the earliest stage of cardiovascular disease, then the risk factor, they are triggering the endothelial cells somehow communicating with monocytes, and the monocytes coming close to the endothelial cells adhere to them, and transendothelial they migrating to them. And I have to say, this good friend of our body later become a problem by digesting the fat part which accumulated inside the intestine. But we were focusing at this part, and there was a question how these two cell types they are interacting. That's why we come up with two questions. If the EV they can carry some message and tweeting some information to the monocyte, and they are causing the monocyte migration. And also for us, it was important to know if these EV are also spreading inflammation to the neighboring endothelial cells. Last 6 years, we were focusing on this type of vesicles, which I call them from now on inflammatory cellular vesicles. And in 2017, we published a paper in Nanomedicine, and we were just focusing on this type of vesicle. And later, in 2018, we discovered their functional impact on endothelial cells and also monocytes. I'm coming back to all of I give a brief result on these two papers, and later this year, into 2020, we published in JEF, and we get deeper to the subpopulation and discuss their content and also their role in inflammation and vascular and monocyte migration. Like many EV scientists, we use ultracentrifugation for isolation of our EV, and we start with the in vitro study, and we selected UVEX as a reference model cell for vascular endothelial cell. We also select TNF-α as a stimuli to induce inflammation in our endothelial cells. We collect two types of EV. We call them UEV, the EV coming from untreated cells, and PEV, EV coming from the inflamed cells. But I remember when we dig to the literature, we find out EV10K also they do some inflammatory job in vascular inflammation. That's why we decided to skip EV10K and collect both pellets of EV10K and 110K using ultracentrifugation. Of course, we wanted to make sure about the purity of our fraction and also to detect a specific marker for inflammatory extracellular vesicles. We applied TM, NTA, and Western blot, and we confirmed the EV coming from this type of cells. They have classical EV markers, including CD63, CD9, and also ICAM1. Keep in mind, ICAM1 is a candidate for inflammation. And we want to make sure this marker is expressed in the membrane of extracellular vesicles. Then later we can use this marker to fish out the subpopulation of inflammatory extracellular vesicles. At this stage, we check the population of EV belong to large and small vesicles, and we find out this fraction is enriched with both of this type of vesicle. And as I mentioned, we were initially interested on developing techniques to check the quality of our fractions. That's why in our lab, we developed surface plasmon resonance, which is a label-free technique. And also in real time, you can see the interaction between the antibody of your interest, which it can be ICAM1 or VCAM1 as inflammatory markers, or CD63, CD9, CD81. This technique is very fast and also single step procedure you can apply to check the surface marker in your extracellular vesicle. And in real time, you can see the interaction between antibody and extracellular vesicle, and you can compare different antibodies. And we published the data in 2017 in Nanomedicine, and we applied it for checking quality of our extracellular vesicles. Of course, these extracellular vesicles, they are packed with other proteins. And also we want to know in vivo system, and dealing with the patient material, they are expressing these inflammatory markers. That's why we work with the acute coronary heart disease and also collecting some patient samples, also healthy control, and we proved compared to the healthy control, our patient samples, they express more ICAM1 in their extracellular vesicles. After lighting these extracellular vesicles, we detect 40 proteins, and we apply here the human inflammation array, and to compare the EV coming from untreated cells to EV coming from treated cells. We find out several pro-inflammatory markers and anti-inflammatory markers in this kind of EV. We confirmed the finding of this array by using ELISA also, because this array is semi-quantitative, and we want to make sure, like TEV, they are carrying some pro-inflammatory markers. And as you can see, they are carrying ICAM1, CCL2, interleukin-6, 8, and CXCL10 and CCL5. These proteins, they are mainly involved in the immune response, and they are chemotaxis factors, which they are, as we said, they are involved with the monocyte migration. This data also published in Frontiers in Immunology in 2018. But so far, as I said, we are dealing with the EEV bulk. Later, I will discuss it. Within this EV bulk, there are some other populations they are existing and which they do the job of inflammation. And at the functional level, we add them back to the UVEC cell line because we want to know if we have this EV, they can also induce in the neighboring cell, and that's why we add them back to the cell and compare them to the positive and negative control. First, we have to make sure these EV are obtaken by our recipients. We have two types of recipients, one endothelial cells and one monocyte. We use UVEC again as a reference model for endothelial cells, and monocyte, we select THP1. After adding back this type of EV, we could see these two types of cells, they react differently to our EV. UVEC cell line, which is endothelial cells, is showing a kind of pro-inflammatory response, while THP1 was showing a combination of pro and anti-inflammatory pattern. Also, if you look, the ICAM was also expressing in both cells. And we want to visualize it. We add them back and we stain ourselves for DAPI for nucleus and ICAM1 as a candidate of inflammation. You can clearly see when we add them EEV compared to TNF-α, which is positive control, TEV is causing the inflammation in UVEC and THP1. If you see here, THP1, which should be round shaped, they are also differentiate to macrophages, but we were not interested to see which type of macrophage, but we can clearly see they are expressing ICAM1 and they are going through some inflammatory behavior. Of course, we were interested to check the adhesion and migration of monocytes. To check the adhesion, we culture cells in monolayer, our endothelial cells, and then we treat it with two types of EV we have, also with positive and negative control. And we send over the monocytes, and we can count the number of monocytes which they are adhered to the surface of endothelial cells. For the migration assay, we use a transfer migration assay, and we put the EV in the bottom layer, and the top layer filled with THP1, and we count the number of cells they can transmigrate from the membrane. Here you can see the results. For the adhesion assay, you can stain your both cells. Here we stain endothelial cells for the nucleus, and also the THP1 with a green. You can see it as a green. The number of cells which they are adhered to the layer of endothelial cells can be counted. We can clearly see, comparing to the positive and negative control, we have a positive response of our monocyte toward our endothelial cells. And about the migration assay, also TEV causing migration in our cells of interest, which is THP1. We can conclude from this part of our result about the EV bulk, which they are containing small and large EV. They are containing some pro-inflammatory markers, and also they are inducing inflammation in both monocyte and endothelial cells. Also, they are causing adhesion and migration in monocytes. But there is still a question: who are the key players in vascular inflammation and monocyte migration? That's why we decide to separate or going for subgroup of EV, and to understand who is responsible for vascular inflammation, who is responsible for monocyte migration. And the result was published in JET recently. You can check the paper if you're interested, but I'm going to briefly discuss the result also here. As we said, we use the ultracentrifugation as our first step of isolation, but this time we don't skip EV10K. We separate it from EV110K, and then manually we add them back together to see the synergetic impact. And again, we check the quality of prep using NTA, TM, and also Western blot. And also we check the content of these three subgroups we have. If you look at the NTA, you can see EV10K, and also if you calculate it per cell, the number of EV10K is half of the EV110K. We can say when cells goes through stress, they release more small EV than large EV. But if you check the protein content, EV10K, they carry more protein. If we calculate the number of protein per EV, we can conclude EV10K protein per EV10K is about 4 or 5 times more. Also, based on the NTA results, we calculate the size range of our EV. We define our EV as a small EV between size of 0 to 100 nanometers, and medium EV between 100 and 200 nanometers, and above that 200 nanometers, we call them large EV. If you look at our fraction of EV10K, majority of EV, they are fallen between 100 to 200 nanometers and above 200 nanometers. It means EV10K contains medium and large EV, while EV110K, it contains small and medium. It means when we use ultracentrifugation, there is a small overlapping in the size of EV. What about their protein content? I'm coming back to them later. But I want to share also these results, which they are very interesting. When we normalize our EV and we load them on Western blot, we could see ICAM is more expressed in the large EV, the EV10K, while the conventional protein markers, including CD63 and CD9, they are expressed more in EV110K. Annexin as a cytosolic EV markers were expressed more in EV10K. Also, we select BAX as a very interesting marker as a negative control because it's expressed in the cells. But initially, we didn't see it in the cells. We are collecting our EV, which is normally we treat ourselves for 24 hours, and then we collect our EV fraction. But the BAX was not expressed. And then when we leave ourselves up to 72 hours under the same condition of TNF-α treatment, they are expressing BAX. BAX is a pro-apoptotic marker. We can conclude our cells at the stage they are producing EV, they don't express or they don't go through like apoptosis, and they don't produce apoptotic bodies. It means our fraction, they are free from apoptotic fraction, apoptotic bodies. And also using the NTA, we could see a cup-shaped EV in EV110K, while in EV10K, we could see a lot of bubbly EV. And when we put them together, we have cup shape and different size of EV. So far, we are just see like this EV. We know them. We characterize them with the NTA, TM, and Western blot. But we claim ICAM is expressed in EV10K. We normalize it to the number of our EV, and we find out the ICAM is more abundant on the EV10K in the larger EV compared to the smaller EV. When we use these different three fractions and we lyse them and we did an inflammation array and we generate a heat map to see who is closer to the parent cell, which is TNF-α treated cells, we find out the composition of protein in EV carrying EV10K or EV10K plus 110K is closer to the parent cell. we can conclude here, like EV10K can be representative of their parent cell, but we are not sure if at the functional level they are like impacting on the recipient cell like stronger. But also, as I mentioned, so far we isolate our fractions based on ultracentrifugation and also centrifugation, but we are not sure if like ultracentrifugation can cause co-precipitation or co-pelleting of proteins. That's why we use immunoaffinity, and I'm suggesting here to the EV scientists, like not relying only on the ultracentrifugation. They can combine their ultracentrifugation with extra step, either mark immunoaffinity or density gradient. First, we decided to use CD9, CD63, and 81 to isolate our EV and look if isolating EV can cause depletion of ICAM. We use a cocktail of CD9, CD63, and CD81, and with NTA we could see the number of our EV after depletion of EV dropped. And when we stain this kind of magnetic beads with the ICAM, we could see a clear like accumulation of ICAM around our beads. And this is a good sign because we knew it's in the membrane of in our EV, but because we use ultracentrifugation so far, we were not sure it's a membrane bound or in the surface of the membrane. And using Western blot and ELISA, we confirm after enrichment and depletion of this fraction, ICAM was gone. We can therefore conclude most of ICAM is packed in the CDS positive EV fraction. This result inspired us to coat beads with ICAM1 antibodies and applying the ICAM1 antibodies to remove ICAM1 from our EV bulk. And we could see with NTA after incubation of beads with EV fractions, the number of EV was going down. And also with ELISA, we confirm it, ICAM positive is enriched, and ICAM negative fraction, they don't have any more ICAM1. Also, the results from Western blot help us to conclude ICAM1 is packed more within the EV population, and it can be a candidate for inflammation subpopulation. Also, we use inflammation array to check the protein content of these 4 types of subpopulations so far we have. We could see like 4 or 5 proteins, they were expressed in positive fraction, and some proteins, especially CCL2 and interleukin-8, was depleted from our fraction. And we knew it so far, these 2 proteins, they do a major job, especially CCL2, which is the MCP1. It's a main protein to induce migration in monocyte. Okay, they are in the depleted fraction, but not in the positive fraction. Therefore, at the functional level, we want to check. Before that, we also have a look to the heat map generated for the protein composition of all these subpopulation. We could conclude the protein composition of the parent cell is more expressed in a large and medium-sized EV, especially the ICAM positive EV. When we generate a PPI to see the protein interaction for these subtypes of EV, we could see this kind of protein which is expressed in the ICANN positive EV, including CCL4, CCL5, CCL10, and ICANN, they are mainly involved in the cell signaling, immune response, and leukocyte chemotaxis. That's why we decided to do some like functional assays. I explained before, for the functional assay, we use monocyte adhesion and migration, and also the ICANN1 expression. We add them back to the cells of interest, which is UVEX, in the serial dilution, EV10K, and then EV110K, and a combination of these two subpopulations. What we find out, both EV, they are inducing in ICANN1 expression, but the majority is induced by EV10K. There was no synergetic impact of these two subpopulations on the ICANN1 expression. And depletion of ICANN or CDS positive or CDS carrying EV also cause the depletion of the diminishing of the ICANN1 expression. It means both sides EV, they are responsible for inducing ICANN1 expression, but it's more related to the subpopulation of which they are carrying either CDF or ICANN1. What about the monocyte migration? The result was very interesting because we could see EV10K, they didn't cause any migration in the monocyte, while the EV110, which is mainly they are small EV, they are all inducing migration in monocytes in a bell shape. And when we use positive control like MCP1, they behave the same. It means they up to certain concentration they are inducing, then the cells they don't respond anymore. But anyway, the number of monocytes passing through the membrane is significantly higher than control. Depleting ICANN1 and also CDS population also could decrease the migration assay. We can conclude here, small size AV, especially their ICANN subpopulation, they are responsible to promote monocyte migration. I would like to wrap up my presentation here with this slide. When cells, endothelial cells, as a major player in the cardiovascular disease, they are going through stress of inflammation. They are releasing a bunch of EV, which is contained EV10K, I will call them large EV, and EV110K, I call them small EV. The number of small EV is much higher than large EV, while the protein content of EV10K is much higher than EV110K or small EV. And I recommend, or our experiments recommend us and everybody, I guess, to use a multi-step isolation of EV to remove some unwanted or unbound or external, we call them external proteins, because we are not sure if they are on the corona of EV, on the surface of EV, or they are co-precipitated. But immunoisolation helps us to remove them, especially CYP2 and interleukin-8. And when we look at the functionality of this EV, they sometimes impact the same, like EV 10K and 110K. They both could express ICANN1 in the recipient cell, in a healthy recipient cell, or in the case of monocyte migration, small EV, they were more responsible. At the end, I would like to thank my colleagues at the University of Hasselt, Maastricht University, and in Vito, which we had a nice collaboration. to answer all of these questions. And if you have a question, I will be delighted to answer.
Well, thank you very much, Baharak, for that wonderful presentation. I'm sure everybody's really enjoyed it. And we have some questions for you that have been coming in the chat box. And I think there will probably be a few that are not in the chat box yet. I wanted to start out though, you know, I'm pretty active on Twitter, and I saw something very interesting that somebody posted recently. And this, I think this was a professor who was teaching a class, and he has his students, you know, present papers, and he asks them questions about the papers, and he likes to have them contact the authors and ask a few questions. So one of those questions was, what for you was the most surprising outcome of this work?
It was about monocyte migration when the EV10K couldn't impact on the monocyte, while the EV110K could. Also, when we use immunoaffinity, we find out CCL2 was gone, while if we look at the bulk EV, CCL2 is overexpressed. And it means besides protein or cytokines, they can be packed in EV. They are containing for sure some mRNA or miRNA, which they are impacting on the recipient cell.
Good. Another question that was asked is, how was the peer review process for you? Do you remember anything in particular that a reviewer asked where you thought, that's a really good idea, I've got to try that?
Yes. It was actually a long journey to publish in JEF, and also it was very challenging. I really liked it because they could pin or could ask very detailed questions, which raising another question in your mind, or designing new experiment for you to make sure what you claim is true. And especially there was a question which at the end we couldn't like clearly answer, was about I guess there is no any paper about the using TM to show vesicle around them after immunoaffinity. I mean, when you stain them, they were asking why the AV are everywhere, but they are close to the magnetic beads, but they are also here. But it can be due to the washing step or due to the TM process you do or preparation. They couldn't answer even like for me still a question. And as I said, yeah, it was a very interesting like challenge for me, and also it could help me to add up some experiments. That's why it took like 10 months or 11 months, and 3 steps of reviewing.
Yeah, I think that's something that's pretty common in JEV, that our reviewers are very thorough. And usually, you know, quite constructive as well, but it can be a long process sometimes. So, yeah. So it's always nice to know that we have, you know, people are volunteering their time to really help us all out with our science, and that's great, because the more the better. So I'd like to go through some of the questions now that are in the chat box, and I'd like to start with Lucia Languino. Lucia, you had a couple of questions here. And I believe you should be able to unmute yourself now. No? Let me unmute you.
Oh, did it. Thank you.
There we go.
Perfect. Okay, thank you. Thank you, Ken, for organizing this great series, and thank you very much, Baharak, for this lovely presentation. Okay, so I used to work on ICAM. That's why I'm more into this field. I used to be in this field. So if I can ask you a couple of things. So imagine when I imagine I come on the EV, I have to assume that it's free to act on other cells, right? But you know that it binds a lot at high affinity, many other ligands. Are the antibodies you have used inhibitory, so you know that you will be displacing the ligand bound to the receptor? Or do you know if ICAM is free to bind other ligands in other cells? And then I have another question. Go ahead.
About the ICAM, it is fine we can release it from the surface of magnetic bit or bind it to another receptor, do you mean?
Barak, I'm not sure I am hearing you well. Could you say it again, please?
Your question about the ICAM, if it's interacting with other ligands, the antibody we use, it was specifically for membrane bound, especially for the membrane bound part of proteins of ICAM. And it was, we tested on Western blot, we can see it's just depleting the ICAM positive one, which especially is interacting with the membrane part of the ICAM.
Okay, we don't know if they are inhibitory of ICAM function. We don't know.
No, we don't know.
Okay, no problem. And then the second question, THP1 are very undifferentiated. And
Yes.
when I use THP1, then I get complaints from the liquid tumor people that they are too undifferentiated. Have you tried this, any of your experiment with another cell line, monocytic cell line?
Actually, with THP1, you have to keep it consistent within the certain concentration. Then if they go above, for example, 10 to the 5, for example, they don't respond or they act differently because they are differentiate. And for us, it was interested to use these cell types because they are not differentiate, because also there was another question, if when they are going through differentiation, if they are becoming macrophage, if they are M1 or M2 macrophage. This is the other question we are now trying to answer them. Also, we isolate the monocyte from PBMC, but it was not within this study, and in other study they respond better than the THP1 to the.
Excellent. Thank you, Barak. Thank you, Ken.
No problem.
Yeah, I was actually thinking of that question too. You know, so how does this change as you have differentiation of monocytes to macrophages? So that's very interesting to hear.
Yeah. Yes, I think it's a very nice subject to work on it and to answer this question, because it's very interesting for many diseases to understand if they are differentiated to M1 or M2, because these two types of macrophage, they act differently in the inflammation.
Sure, for sure, yes, indeed. So, well, now let's move from Lucia to Lucas. Lucas had a question, a technical question here. Lucas, are you there? Let's see. Yes, let me see. Okay, I will just go ahead and ask this question. So it's during the isolation, do you wash the 10K EV pellet? So I know you showed that you washed the 110K, but do you wash the 10K EV pellet? And do you think that that perhaps there's a lot of extra, let's call it extra vesicular protein?
Yes, we do for all the functional assay, and even like protein profiles, we have an extra washing step with the PDS.
Okay, all right, that should answer that question then. Now we have a question from Tom. So my co-host, Tom, go ahead, Tom.
Hi, very nice talk. I was wondering, you are now one of the several people that show that EVs can contain cytokines. So how do you think these are associated? Are they on the surface or can they be within the EVs? And can these be part of the protein corona, or what are your thoughts about this?
Very interesting question, because it was also a big conversation between me and reviewer, because initially we claimed like CCL2 and MCP1, they are in the corona, because after depleting with the immunoaffinity, they were gone. But we are not sure about it. There is a paper, I don't know it by heart, they use proteinase K, for example, to shape EV and then check the surface proteins, and or using different way of lighting EV to show if the cytokines are inside or in the membrane. There is a different protocol you can apply and answer this question. It was a nice paper, but unfortunately they use a kit for isolating EV, which in my opinion, applying like this kind of kit just isolating everything and just applying this kit cannot answer this question. I think using protein escape and different types of lysis buffer, it can answer the question.
All right, thank you.
So I guess a follow-up to that. So Pasquale D'Acunzo, do you want to go ahead and ask your question? Because I think this is about different methods that we might apply and what is stringent enough to say this is corona versus this is otherwise loosely associated. Pasquale? You might be having some unmuting issues here. I'm not sure why unmuting is not working. But in any case, I'll just follow up here. Pasquale asked about a density gradient. So have you done density gradients here? And do you think that a density gradient would give you a more stringent result than, for example, immunoaffinity? Like, what would you, if you had to choose something, another technique to try, what would it be?
Very interesting question, actually. Also in our lab, we optimize density gradient and mainly for the plasma samples. The problem with the density gradient is if you want to do functional assay later on, recovering the EV fractions from density gradient is much more difficult than the immunoaffinity. That was our impression, because we are currently doing this experiment with the density gradient, but not for ICAM, for other markers, inflammatory markers, but adding back the fraction of density gradient sometimes also purifying it from the density gradient is harder, and especially for the functional assay. But it's an option to get rid of the impurities.
Yeah. I'm wondering too, because you found that there were some cytokines that were associated with EVs with some of your subtypes, right? Whereas others were not. So, for example, if I recall correctly, it's CCL2, IL-8. You were not finding those in the ICAM positive subset. Have you looked at the properties of these proteins? Is there something about those two, for example, that sets them apart from others?
Why they are in the corona or outside? That's a good question, actually, to look to the configuration of this type of protein, why they are outside of the EV, not packing inside. Maybe it's dependent to the pathway they are expressing. They are just expressed on this outside, and then when EV are coming outside of the cells, they are just around the protein corona EV. But honestly, it's a good question, and I will write it down. I will look it up. Why CCL2 has a major protein in migration of monocytes? It's just it's not packed within EV.
Okay. Yeah. So that also about subsets, we have Tina O'Grady. So Tina, I'm going to see if I can unmute you, because I think your question is important too. Okay.
Is that working?
It works. Okay.
All right. Thank you for the talk. But my question was, so you present these as two different distinct populations at 10K and 110K, and you certainly show that they are different, but I'm wondering if you think these are really representative of two actual sort of biologically distinct populations, or if maybe it's more of a spectrum that you're picking up one end at 10K and one end at 110K, or even something more complicated than that.
Very interesting and challenging question, but I can say, because you could see in NTA there's a spectrum in the size of this kind of vesicle. When we use EV10K, it's not exact 100 to 200 nanometers or 410K, I mean, but there is a spectrum in the size. What I want to suggest to ISEF, we have 10K, we have 110K. In between, there should be one extra step to isolate because as I showed, in medium size, they are overlapping. If we can isolate this medium size, like with 50K, for example, and exclude all these three, we can cover the spectrum of extracellular vesicle released by cells when they are going through stress.
Cool, thank you.
Very interesting. So maybe our lives are even going to be more complicated than they already are.
Yeah.
Let's see. We also have, we have a question from Tanina. Tanina, are you able to unmute? I will try to unmute you if not. I don't know why we're having a real problem with unmuting today. There we go.
You succeeded. Good morning, everyone. So it's impressive actually, and a very nice work. And I was wondering what will be the next step in this study from your perspective. Where do you want to lead this very promising finding?
If I heard it well, you asked about what will be the next step in our study.
Exactly.
Yes. So far, we proved ICAM positive subpopulation. They do the major job. We checked the protein content, but we didn't check the RNA content. Also, we work in vitro, and we didn't apply it on the patient material. We know this subpopulation can be a candidate of inflammatory extracellular vesicles. We can use the same concept for patient material and check if they are carrying a package of markers which can be applied for early diagnosis of a disease like inflammatory disease, which for us, cardiovascular disease was our main focus. I hope I answered your question.
Good. Okay. Very nice. And now a question from Alex. Alex.
Yeah, I'm sorry, I don't have a camera. Can you hear me?
Yes, we hear you. Thank you.
Oh, great. Yeah, I was thinking about the possibility of using some kind of density gradients to better isolate the subpopulations, and you were concerned about purifying the vesicles after the density gradients to take away the media. So, because the media, the density gradient media, were interfering with your assessment, I was wondering what kind of density gradient media you're using. Because in our experience, IDX is mostly neutral. Low concentration of IDX is mostly neutral in most in vivo and in vitro assays. So you really do not have to do extensive purification of this. So that I was wondering what is your density gradient media that you're using, but you have to do so much purification.
Thank you for your question. Because first, the question we wanted to answer, it was about inflammatory extracellular vesicles. We want to make sure we are isolating the ICAM1 subpopulation, which can be a candidate for inflammatory extracellular vesicles. We use immunoaffinity. With density gradient, you cannot isolate certain subpopulation like ICAM1 positive. With density gradient, it's like kind of like ultracentrifugation, which is based of speed. There, based of density, you can like purify your EV fraction, but it will be a bulk again. It is not a. It will be based on the density, different subtypes, but it is not related to the inflammations.
So if I understand correctly, then, and sorry to interrupt, but it's not that you think that the iodixinol itself is interfering with detection of the ICAM, rather that the population is too low, you don't have sufficient recovery. So it's not that you think that there's something about the gradient itself that is interfering with detection.
The gradient depends the type you use. The surface gradient, it can interfere, which we tried it, and also. As I said, we were interested in the inflammatory extracellular vesicles. That's why we decided to use immunoaffinity.
Thank you. That was clear, yeah. I mean, the way Kenneth put the question.
Your sound is very far. Can you just...
I'm sorry?
Your sound is very far. Can you talk a bit louder?
Oh, yeah, no, thanks so much. It's clear now.
Sorry.
Okay, good. So that's clear. So I actually, I know this was not the focus of your talk today, but we haven't had very much exposure to surface plasmon resonance in the Journal Club, and you have a considerable experience with that. So could you just tell us a little bit more about how you use SPR, which platform you use first of all, and what other platforms are available?
Yes. Surface plasmon resonance, the one we use, it was C200, which is a label-free technique. You need to immobilize your antibody on the surface of a gold chip. This gold chip also functionalizes with a carboxyl group. We have different types of chip, CM5, C1 chip. We select CM5, which is carboxylated surface. Then your antibody will stand like this on the surface, and then in real time you can send over your EV and see the interaction between antibody of interest and your EV. Especially the surface marker. You can also digest your EV and send it over to check inside EV, but we were interested to see the surface marker, which is what ICAM and also CD9 and CD63. It's a very fast procedure, and also like for Western blot, you need to 2 days of work to get to the results. Sometimes it's not working. It's very like direct method. You can see in real time the interaction between your EV and your antibody of your interest.
And would it be possible to use that technique to assess something like what Lucia was asking about? So for example, if you are able to immobilize your EV, would you be able to look at binding of ligands, for example, to a receptor on the EV, or is that beyond the, is that happening too far away from the surface, I suppose?
No, it is possible. Actually, you can do the kinetic study and to check the affinity of one EV toward one or two antibodies. But the problem, initially we had this plan to capture and to do some kind of isolation on the surface of SPR. It means you send over just your conditioned medium or plasma, and using CD6, CD9 to capture your EV and sending over the secondary antibody to see what is on the surface. But the channel on the T200 was too narrow to have two antibodies at the same time. That's why we start our collaboration with the Fox Biosystem. They have deep SPR. They have this kind of needle types of chip, which from one well, it is detecting your EV, and in other wells, it can interact with other solution which is containing the secondary antibody, and you can see you have your EV and you have this specific protein on the surface of your EV. But it needs optimization.
Yeah, of course.
But results for the T200 and applying SPR, which is very nice and fast forward techniques, is published in Nanomedicine, and there are a couple of nice papers related to the SPR. I think we have to introduce also this platform to the ISEP, but the problem is it's a bit with the chip we use is kind of expensive, but we optimized the chip. You can regenerate it up to 50 times. It means the antibodies stay on the chip, and you can send the next sample, and you can see the interaction of your EV with the antibody and regenerate it, and again send over new samples. And it was possible up to 50 times.
Okay, so Lilia, go ahead and unmute yourself because you have a follow-up. But as you're unmuting, Baharak, what do you mean by expensive? How much is that? That's a ballpark.
Because it's a surface of gold chip carboxylated, and you have to buy it. Compared to the Western blot, it's kind of expensive.
Okay.
But as I said, we can just optimize it and for regeneration and use it 50 times.
Yeah, no, that's good. So Lilia, are you there?
Can you hear me?
We hear you. Thank you.
Yeah. Yeah, thank you. I'm really interested in this technique, and of course, I think I'll go deeper in your publication, which you actually showed us today. And I'm just really wondering about the sensitivity of that technique. Have you tried to compare some ELISA assays, for instance, like the sandwich ELISA assay with this technique, and how is it sensitive if you're working with a really low amount of material? For instance, if in the perspective you will isolate the vesicles from the blood and you want to be, you know, in somewhere around like out of 1 ml, and how do you think, is it actually more sensitive or not comparing to ELISA, or you haven't tried that? Will be amazing actually to have the chip with a lot of antibodies at the same time and in the moment just detect them all.
Of course, comparing these two techniques is difficult because this is a label-free technique. You don't use any label or secondary antibody, which is labeled. It's a direct detection. And normally for biomedical science or people which they are dealing with new drugs, they use SPR to check the sensitivity and specificity and also the affinity of their drug. And SPR is now to check all the sensitivity of your test. Because ELISA and other techniques, you use a secondary antibody, you are boosting or enhancing your signal maybe 10 times more to detect, but SPR, you don't enhance your signal and it's just directly you detect the interaction of your ligand and analyte. Of course, when you deal with EV and sometimes you have 100 microliter, it depends the concentration of EV. But with the dipstick Foxfire system of SPR we have, with just 100 microliter of samples, you can just nicely see the interaction between EV and surface.
Okay, thank you. Thanks.
Well, we're coming to the end of the hour, but we have one more question, and so I'd like to ask Raquel to please ask this question about toxicity.
Hi, thanks for your talk. So my question is regarding the EV concentrations that you use in your assay. Do you have a rationale behind the concentrations that you use? Do you think using higher EV concentration would actually cause any toxicity? Or would be a plateau in the effect on the other cell types? Thanks.
Good question. Actually, the rationale behind the concentration we use is explained now in my paper. But what we did, we worked with the volume base. It means if we deal with the 15 ml of conditioned medium and we isolate them and we put them in 1 ml, we calculate how much we can add them back with the same number of EV coming from 10 to the 6, for example, cells to the cell of interest. And then below that one, that's the highest concentration we add. We make a serial dilution and to see if with increasing the number of EV as recommended by myself, to work with a serial dilution, and we make a serial dilution below that volume or concentration. Of course, we were concerned about the toxicity of extracellular vesicle, and we performed like an Annex-5 assay to check if the cells are going through the apoptosis, and we find out EV, they are for both cells, not monocyte and endothelial cells. None of them, they didn't go through apoptosis or stress coming from the EV using the Annex-5 assay.
Okay, thank you.
You're welcome.
Well, thanks to everyone for joining today, for the great questions, and most of all to you, Baharak. Thank you for sharing your knowledge and experience. It's been a wonderful hour for me, and I've learned a lot. So so we will be archiving this presentation on YouTube, and if you'd like to subscribe, just search for Extracellular Vesicle Club on YouTube, and you can get the updates. So thank you, everybody, again, and I hope you have a wonderful rest of the week. Talk to you again soon. Bye now.
Bye.